So jumping into the case, we have that presents with a progressive white lesion encroaching on the cornea in the right eye. Could I have one of my co-residents describe the image to me, please?
Dr. Aldred?
All right, so we're looking at slit lamp photos of the right eye, starting with the lids and lashes, maybe few telangiectasia, some MGD, but otherwise no matarosis. The conjunctiva, attention is drawn temporally to this large lesion that's extending onto the cornea. On the cornea inferiorly, you can see some leukoplakia. Superiorly, the lesion extends and you can see the vessels throughout, a little bit of corkscrew, not growing completely to the leading edge of it. And a little bit irregular. In terms of the anterior chamber, looks formed, no obvious cell, some trace NS. Otherwise, iris looks normal.
Excellent. So yeah, we see this large multilobular mass with scalloped borders extending onto the nasal cornea. There's leukoplakia, and the vessels you see don't extend all the way to the margin of the lesion. This patient presented with an otherwise normal exam. His vision was 20/20 OU, pressure was normal, he didn't have any APD. Differential for these sorts of lesions is broad, but just honing in on what we see typically, it could be a pterygium, it could be an amelanotic melanoma, OSSN, papillomas, or panis are on the differential as well. What would be the next steps?
I'd want to start with an anterior segment OCT.
That's excellent. That's exactly what we did. Could I have you briefly interpret the findings?
Yeah, so centrally, you can see there's this sort of thickened, hyperreflective epithelium. And then towards the left, you always want to look at the transition point. And in this one, it looks like it's a pretty abrupt transition point from the normal epithelium to that sort of thickened area.
Perfect. Yeah, we see that sharp vertical transition point between normal and abnormal epi and the hyperreflective thickened epithelium like you alluded to. So given the clinical history and findings, the diagnosis of ocular surface squamous neoplasia was pretty comfortably made. In this case, the lesion demonstrated a lot of the typical findings of OSSN, and the OCT also had a lot of the classic findings that we look for. However, this is another case of a patient with a little more subtle findings. It was referred for an inflamed pingueculum, treated on topical steroids for some time without much improvement. On the surface, it looks like there's not much to see. However, honing in on the lesion, we see this abnormal vascular array and kind of a pinwheel configuration. The ANSAG OCT of this patient also demonstrated a hyperreflective thickened epithelium, but if we look at the transition point, we don't see that sharp 90-degree cutoff. We kind of see a sloping edge to it. So the diagnosis in this case wasn't really clinched. So what do we do in situations like this where we don't necessarily know what we're dealing with? Obviously, the gold standard is to get tissue and do a biopsy and look at it under the microscope. However, there's been recent trends towards non-invasive diagnosis methods. For example, with the advent of high-resolution OCT imaging, we saw that the transition point, thickened epithelium, and hyperreflective epithelium are the three key findings that we look for, and the sensitivity and specificity has been quite high in recent studies. However, what about in cases where you don't have access to an OCT, or you can't interpret the OCT, you don't have training in it, or maybe the lesion is very subtle, or the ocular surface has a lot of other comorbidities like SJS or AKC? In these situations, then, we point towards the work of Dr. Pelez and his colleagues who are addressing this very question. So, Dr. Pelez, please.
Thank you so much. So yeah, I'm going to give you a brief presentation on what's been going on in the lab in terms of development of liquid tear biopsies or diagnostic panels, or to develop diagnostic panels for some of these ocular surface diseases. So liquid biopsies, this is going automatically. Liquid biopsies refers to the sampling of bodily fluids or bodily compartments. Where in the eye, we have several amenable compartments, both the aqueous and vitreous humor, as well as the tear film, that can inform us as to the underlying pathologies. And then when you sample liquid biopsies, you have to decide what molecular compartment you expect to find differential signatures that will allow you to have some diagnostic power. And these include most of the biomacromolecules like lipids, carbohydrates, proteins, and nucleic acids. Now, when developing an assay, you have to take into account the limitations of your sample. In this case, we're dealing with mostly acellular fluids in the tear film that are very low volume, which lends itself to more nucleic acid work. So through PCR, we can amplify whatever molecular signature by several billion-fold through PCR, and that gives us a good readout or a good sensitivity to read out what the molecular signatures are. So we focused on RNA because we don't know the underlying mutations or genomic aberrations that we're looking at. And when people think about RNA, traditionally, they think about the central dogma in molecular biology of DNA to RNA to protein, which refers to the coding RNAs or the messenger RNAs. So the problem with messenger RNAs outside of cells is that they are highly immunogenic. They're one of the most immunogenic molecules that we have, and they get very quickly degraded. So within minutes, you lose most of the mRNA signature, and that led to the development of modified RNAs through things like N1-methyl pseudouridine, which then enabled the development of the mRNA vaccines that we saw for COVID, and now RNA therapeutics, and won Weizmann and Carrico their Nobel Prize. Now, on the other side, there is a whole host of other RNA species that people aren't as familiar with. There are non-coding RNAs. So these include things like housekeeping RNAs, the ones found in ribosomes that link messenger RNAs to the ribosomal translational machinery, as well as interfering RNAs, which have a role in guiding ultimate cell phenotype. The latest one being awarded a Nobel Prize, which was last October, was for the discovery of microRNAs by Ambrose and Ruvkun, which is the focus of our work. And why is that? Because we looked at the RNA profile in tear samples that we had collected here at Bascom Palmer. And while we have several samples that have a good profile of RNA, and just for those of you that aren't familiar with these, what you're looking at is a size versus quantity distribution of RNA species. And the two shaded peaks here relate to the 18S and 28S ribosomal RNAs. So this is the area where messenger RNAs would be found. So these two samples would be amenable for messenger RNA sequencing. However, most of the samples that we collect from tears look more like this, where you see that there is really no messenger RNA molecules there to assay. However, if you can look at every single sample has a very nice, consistent and stable peak for microRNAs, which bolstered our knowledge or our idea of pursuing microRNAs as the molecular compartment that we would be sampling. Now, microRNAs, as I mentioned, are regulatory RNAs. So they're short hairpin RNAs, which contain a particular sequence that binds to target genes to either block their translation or cause their degradation. And that's how they affect ultimate cellular phenotype. So just like the normal transcriptome through messenger RNAs, microRNAs affect ultimate phenotypes and disease processes or normal processes as well. MicroRNAs are stable enough that they've been used by several labs in blood liquid biopsies for both diagnostics as well as staging of different cancer types. So we said, all right, why don't we look at the microRNA profile in tears, since we have a very nice peak, and see whether a signature in there can help us distinguish what would be OSSN or malignant versus benign lesions of the ocular surface. So this is our program right now. It's a robust program looking at different indications using tear films for what we believe would shed molecular information to the tear compartment. And the lab and clinical trainees, as well as our collaborators at Bascom Palmer, everybody being very dedicated to the project. And I'm going to be talking about, for the purposes of this talk, about our work with Carol Karp on development of diagnostic panels for OSSN versus pterygia. So we started off by doing next generation sequencing of 10 proven OSSN samples, 10 proven pterygia samples, as well as 10 healthy controls with no underlying pathology. We obtained microRNA differential signatures, which gave us some biology or insights into the pathobiology of what causes malignant transformations in OSSN. And then from this signature, we decided to develop a simple PCR assay to validate a panel that would allow us to collect samples run them through PCR and stratify patients as to whether they are at risk for malignant transformation in their lesions. So just to go a little bit into the molecular pathways, we saw that the enriched pathways for the target genes of the microRNAs that we were finding were related most heavily to pathways in cancer. So that obviously falls in line with what we're seeing. We're comparing OSSN versus pterygia here. But it also gave us some insights as to what molecular events or crosstalk in pathways may be actually associated with malignant transformations in OSSN. And this is very recent finding in other tumors where we see that the RAS, PI3K, and Hippo pathway crosstalk with each other in mediating malignant transformation of neoplastic lesions. But more importantly, we then decided to move into the development of the PCR assay. And for those not comfortable or not knowledgeable about how you develop clinical assays, there's a lot of validation that has to take place. And one of the things that's challenging in microRNA PCR signaling is that there are no normalizing genes. So when you run messenger RNA for gene expression signature, there are several RNAs that are considered very constant and housekeeping genes that we can use as normalizing genes. There's no such thing for microRNAs. So one of the first challenges that we faced was finding control microRNAs to normalize tier samples. So by analysis, using an algorithm called Norm Finder, we compared control as well as pathological samples for the most stably expressed microRNAs in all tier samples throughout all indications, as well as in healthy controls. And through iterative testing, we ended up with 2 control microRNAs that seem to be capable of normalizing microRNA signatures in any tier sample, in any human tier sample. Similar on the pathological side, we started off with 72 differentially expressed microRNAs between OSSN and pterygia. And then through subsequent iterative testing of incoming samples, we curated that down to 10, then to 6, and finally to our final panel of 4 differentially expressed microRNAs that give us diagnostic power to distinguish OSSN versus pterygia. So this is our panel as it's designed so far. We have 4 microRNAs that are differential between OSSN and pterygia, 2 microRNAs that are upregulated in OSSN, and 2 that are downregulated in OSSN. And these are normalizing microRNAs for human tiers that can be used across any indication. Now, something to take off of this slide is that if you use any one of these markers individually, you get roughly 60% diagnostic power at distinguishing malignant versus benign lesions. However, if you combine them into a panel and use the readout for the multifactorial or multivariable readout, then your diagnostic power increases to 94%, and it's very significant. This was work done by Acadia Moyer-Sims, a previous PhD student in our lab. She presented this at several competitions, and she won both the university-wide as well as the statewide 3-minute thesis competition for her work in the development of this OSSN versus pterygia panel. So ongoing work on diagnostic panels out of tiers. We're validating the microRNA panels and signatures for all the other indications that I showed you. This goes into obviously sequencing additional samples, validating the PCR parameters for each probe, and running some mass clinical studies where we get samples in the lab where we don't know the final pathology. We make the molecular diagnosis, and then upon unmasking, we compare our notes to actual histopathological diagnoses. And then we have a robust program right now with our Office of Technology Transfer to partner with commercial entities to go through the regulatory and validation steps to get these clinically approved, at least for the OSSN versus pterygia panel, so that we can translate them from the bench to the clinic. The take-home message of this research is that everything in our bodies, every cell and tissue has a normal or a specific molecular signature. And if you know where to look, so if you know what the appropriate sampling fluid or tissue is, and the molecular space that you expect these differential signatures to occur, then you can design some of these panels or find some of these biomarkers that can be very informative for both diagnostics, prognostics, or just general understanding of disease processes. And back to our patient.
Thank you very much, Dr. Perez, for that exciting work. Going back to our patient now, just like there's been a trend towards non-invasive diagnosis methods, there's also been a trend in recent years towards non-invasive management of these lesions. Work done by our very own faculty members here compared a survey of providers in 2024 to earlier years and found that there was an increase, a shift in the trend of providers who are using topical management first. Concurrently, there's also an increase in the amount in the frequency of each topical agent being used now as well. This graph visually demonstrates those trends. We see that excision alone approaches have recently fallen out of favor, and excision with postoperative medical therapy has increased in frequency in recent years as well. Our patient was treated with 4 cycles of topical 5-FU, and we see in the before and after pictures that there's been a near complete regression of the lesion. Comparing the OCTs from the first touch point to the follow-up visit, we also see that there's been almost a complete normalization of the corneal epithelium as well. So in conclusion, our diagnosis of OSSN has been revolutionized by the advent of these non-invasive modalities like OCT and tear film assays, like the work that Dr. Perez and Dr. Karp are doing. And in parallel, we see trends towards non-invasive management of these lesions as well.